Viruses as vector for gene transfer. In case of plants, we have also focused that some viruses, they have the natural ability to infect any cell types. Again, if we focus on the examples of some bacteriophages, कि कुछ बैक्टीरियोफेज ऐसे होते हैं जो कि बैक्टीरिया में अपना जीनोम इंसर्ट करते हैं और फिर उसकी जो मशीनरी है उसको ऑक्यूपाई कर लेते हैं और फिर अगर आपको याद हो कि वो लिटिक साइकिल में जा सकते हैं या लाइसोजेनिक साइकिल में जा सकते हैं. सो इन दिस वे दे कैन प्रोपगेट by using the host bacterial machinery. In the same way, we also discussed in last topics that bacteria also have the ability to transfect a number of plant species so that after adsorption, it can inject its DNA into the host genome. Similarly, this ability of the viruses, it can also be exploited for the transfection of animal cell lines. और इसके अलावा बहुत सारी वायरल इन्फेक्शंस भी होती हैं ह्यूमंस में या एनिमल में, तो उसमें भी वही स्टेप्स होते हैं कि एड्सॉर्प्शन हुई, डीएनए इंजेक्ट हुआ और होस्ट सेल के अंदर जाके वायरल पार्टिकल्स जो हैं वो रेप्लिकेट करते हैं और इस तरह समटाइम्स सिस्टेमिक इन्फेक्शन भी हो जाती है जो कि पूरे टिश्यू में या बॉडी में स्प्रेड हो जाती है. सो दिस एबिलिटी आई कैन से दैट it can be exploited to transfer our gene of interest. And nowadays, variety of viruses have been developed that can be used for the purpose of gene therapy, that for the cure of different genetic diseases. This ability of viruses has been used. And in addition to that, the variety of vaccines have also been developed by using or by synthesizing recombinant viral particles. Now, in this case, if you remember our discussion related to some vectors that are based on bacteriophages, that how we can replace our foreign or exogenous DNA. That some portion of the virus or the viral genome, it is non-essential, that it is not required for packaging, so it can be replaced with exogenous DNA. And sometimes it has been reported that most of the genome it has been replaced, and then after infection it can deliver our gene of interest. And here I can quote the example of adenoviruses that are DNA viruses having linear double-stranded DNA. It has been used to synthesize different type of derivatives that can be used as a vectors. Now, what is the reason that adenoviruses can be used as a vector to transfect animal cells? Because it contain different characteristic features, like here the transformation, it is comparatively stable, and large amount of DNA it can be delivered, and efficiency it is very high, that is the efficiency of plaque formation it may be 10 raised power 11. So transformation efficiency it is high. So because of these reasons, adenoviruses have been favored for animal transfection. Like bacterial transfection, here we also have to take into account that the modified viral particles that we are going to use as a vector, they should not have any biosafety issues, that they should not induce the infection into the host organism if we are going to use them in vivo. So Overall, we can say that they are very important, and some vaccines even have been developed by using recombinant viral particles.